Okay, well then, welcome everybody. This is the first BioGeoSCAPES Modeling Webinar. We are really excited that so many people are joining us today, and we have two great talks to look forward to. But before we start with the presentations, I will give it a couple of minutes to introduce BioGeoSCAPES and the webinar series. So, BioGeoSCAPES is a global research initiative. It has been launched in the mid-2020s, and the aim is to improve our understanding of ocean microbial biogeochemistry. So we are interested in how microbes in the ocean influence the cycling of elements and how these processes affect marine ecosystems and the global climate system. And of course, any type of feedback between these players. And central to BioGeoSCAPES is promoting international collaborations. So I love that we have many participants registered from African, Asian, and South American countries. And we are, of course, glad that you are here and also from everybody from other continents. And key goals of BioGeoSCAPES are developing microbial maps on a global scale. These will then show the distribution of microbes and their genes, and also foster developing a new generation of ocean ecosystem models that will integrate these great new omics data. And this webinar series and also the upcoming workshop focus on these new modeling approaches. So really, BioGeoSCAPES focuses on processes of very different scales, starting from very small intercellular processes, such as how cells allocate energy and resources or how they form proteins, and zooming out to the level of organisms or group of organisms, working on questions such as how are marine microbes distributed, how do they interact with each other, how can we make the best use of these limited observations that we have. And then zooming out even further, how do microbes affect ecosystems and climate on a global scale? And the models that are used within the BioGeoSCAPES community reflect these different scales. They are, for instance, models focusing on intracellular processes. There are statistical approaches, how to understand or extrapolate data on organism level, and there are global Earth system models. And it is, of course, an important question to us how these different modeling approaches can be integrated with each other or synergized in some way to learn something new. So our goal with this webinar series and also the in-person modeling workshop is to connect modelers from different groups and figure out ways how to best work together to achieve the BioGeoSCAPES mission. We are very much aware that it's also crucial to connect modelers and experimentalists to make sure that we are using the new observations in the best possible way. And the plan is to also have a future workshop for that. But the current workshop and the webinar really focuses on collaborations between modelers. So this is a quick advertisement for the following webinars and the workshop. Today, we will focus on global biogeochemical models, and we have two great presenters that I will introduce in a minute. 1 week from now is a webinar on statistical approaches, and the last week or the week after is about models that operate on cellular scales. You can use the QR code to register. Obviously, everybody who sees me right now is already registered, and the registration works for all 3 webinars. And then the in-person workshop will be in September 10 to 12 this year at Hui, so that's 2 hours from Boston. This workshop will be co-hosted by CComp. And the application for that will open on May 27th. We will send out an email to everybody who is registered for the webinars. Now, one last thing before I introduce the speakers. We have a code of conduct, and these things are important, so I will read it out to you. BioGeoSCAPES does not tolerate harassment or discrimination in any form, certainly no physical or verbal abuse, and also no deliberate disruption of presentations. Please just be respectful. We love to hear your constructive criticism and ideas, but don't personally attack individuals. And please be an ally. If you witness troubling behavior, please report it. I know this can be difficult. And I really think science is best if we listen to each other, so let's be kind. So, today, the format is as follows. We will have 2 talks today. Each talk will be 20 minutes long. And after both of the presentations, we will have a moderated discussion. You will have a Q&A button at the bottom of the screen, and you can use it to submit questions already during the presentation. And we will read out those questions, or as many as we manage, at the end of both presentations. So our first speaker today is Dr. Pearse Buchanan. He is a research scientist at CSIRO in Tasmania. He develops the Australian ocean biogeochemistry model and investigates how the ocean is changing in response to climate change. And he's currently particularly interested in the nitrogen cycle. I also hear that he's a passionate dancer and even a teacher of Cuban salsa, so it's surely great to hang out with at conferences. And Pearse, we are so glad that you're with us today. Please, floor is yours.
Sorry, what? I missed that last part? Am I going ahead? Okay, great. Okay. Okay. Hello, everyone. Right. Are you seeing the presenter? Are you seeing the slides? Yep. Okay.
I can see the slides, yes.
Great. Hi, everyone. I just want to thank the organizers and greetings from Tasmania, the island off the bottom of Australia. So I'm very excited to talk to everyone today about something that fascinates me, which is the intersection of microbial metabolism and biogeochemical modeling, and particularly its relevance for the nitrogen cycle. So I think one of the reasons it fascinates me so much is because um, I just love the contrast of how these tiny microbial engines scale up to affect global biogeochemical cycles that impact the climate, right? So this is a great figure from Emily Zackham in a paper in 2020 showing a bunch of different metabolisms there, from photoautotrophs to nitrifiers to methanotrophs, and all of these things all these metabolisms are executed by the tiniest cells, and um, there are millions of cells per millimeter of seawater, so it's easy to see why these tiny engines scale up to affect things on the grand scale. When you integrate this through to the area of the ocean and then 1 meter of seawater, you find that there's more bacteria in the upper meter of seawater than stars in the observable universe. So you can see why these things are important and why these metabolisms have such a big impact on the global scale. And so I think the challenge for us as a community is to link the micro with the macro. And I guess I'm preaching to the converted here. We all know this. And in my mind, maybe a more specific way of talking about this is to link metabolic ecosystems with their environment, both on a micro scale, but also on the basin scale. And so we have to try and understand why maybe a community looks different in the North Pacific to a community that looks different in the South Pacific to something that looks different in the equator, and each of these differences in the ecology of these areas will have an effect on the cycling of important things like carbon dioxide. And what we do to link the micro with the macro, we use biogeochemical models. So what is a biogeochemical model? Well, a biogeochemical model is something that links the physical transports with the ecosystem. So we discretize the system, right? We represent the ocean in many boxes. We put the boxes together, and we have physics routines within them that transports fluid and traces like heat and salt. Also things like oxygen and other biogeochemical traces. And then we have an ecosystem component as well, which within each of these grid cells that make up the model, we have an ecosystem component where, for instance, here, nitrate is being consumed by phytoplankton, which is then being consumed by zooplankton, which is then being consumed by detritus, and eventually being consumed by bacteria to recycle that inorganic nutrient from which it came back to what it was. And the way that we represent this system, these metabolic fluxes within the ecosystem model, is very much important. Oh, I forgot to start my timer. Here we go. Okay, timer's on. So how we represent these ecosystem fluxes is important. We can represent them simply. We can represent them in a more complex way. Much like we can represent the physics in a simple, coarse resolution to a complex way, finer resolution, where you start representing things like sub-meter scale filaments and eddies. And each, whether it's physically complex or biologically complex, it's going to have a tangible effect on things that we care about, like carbon dioxide, right? So how we represent these models is really important. They're very powerful. They scale the micro to the macro, but care must be taken to represent the ecosystem component well. And this is where I think BioGeoSCAPES really comes in. I think the kinds of studies that are done under the umbrella of BioGeoSCAPES really gives us the care we need in order to move forward and adequately represent these ecosystem components within biogeochemical models. So that's my perspective. And I want to dive a little bit more into how we represent metabolisms. So each of these processes here, phytoplankton, photoautotrophy, and heterotrophy in the bacteria, their metabolisms and how we represent them is important. So I think about the metabolism as being a combination of an oxidant and a reductant that is generating electron transport chain that then generates ATP to create biomass. The microbes grow, they die, and they excrete products. And so in this example of heterotrophic metabolism, we have the oxidation of organic matter, and that produces an electron that is then accepted by the oxygen. This generates ATP, some number of ATPs. This is the catabolic reaction that then provides the energy needed for anabolism, the energy demanding process, which creates organic matter. And we can represent this in a spectrum of approaches, okay, from simple to complex. The important thing to take away is none are wrong and absolutely none are right. It's a horses for courses, as Al always says, situation. And so we're going to start with the simple, and I'm going to categorize them into three main camps. We're going to start with rates. This is the simplest of the approach, right? Where we don't have any biomass, we just have the oxidant reductant, and we excrete the products. This is where biogeochemical models started. We have organic matter, in this case, for heterotrophic, taking our heterotrophic metabolism as an example. We have the oxidation of organic matter, the consumption of oxygen, and the production of CO2 and H4. That's it, right? So that's the sort of rate approach. And we can make it a little more complicated, more interesting by adding things like a temperature effect, an oxygen effect, maybe the amount of resources required will affect these rates. We can then scale it up in terms of complexity to traits. So traits now, it's the same, except now we've added an explicit biomass. This is how we represent phytoplankton and zooplankton in our models today, mostly. So in this sense, we just add that anabolic pathway where we create biomass and the biomass dies as well. So it grows and it dies. And then finally, we have fates. And I'm not sure if this word fits, but I'm going with it because it rhymes. But fates is where we then people have started to play with adding in cellular scale, genome scale models that within the biomass itself. So in this case, we have things like protein allocation variations in response to our environment changing, right? Maybe temperature increases, maybe that increases ribosomal translation efficiency, maybe the stoichiometry changes because the cell doesn't need as much phosphorus. That's an example of where one of these models might come in to feed back on, say, important traits like growth rate or maybe the metabolites that are produced. This is certainly not my area. And so I'm just pointing to some studies that are published in this area. Certainly not exhaustive, but I would encourage people to go and look at these studies if they were interested. And I think each of these approaches is right for different questions. So if you're just interested in how the environment affects the cycling of, say, CO2, Maybe you only need rates. Maybe an important question there is how does temperature affect carbon export by altering mineralization rates? That could be answered using just this simple approach. If we scale up the complexity and go to traits, maybe we're more interested in competition and synergies, mutualism between different classes of microbes. So this is where functional diversity might affect cycling. A good question here is how does competition for ammonium determine the distribution of phytoplankton or ammonia oxidizing archaea, for example? And then for fates, maybe we're more interested in metabolic adaptation in response to environmental changes. So, good example, under iron limited conditions, how do diazotrophs allocate energy between nitrogen fixation versus respiration, for instance? And really today I'm going to talk about this middle approach, which is what I use. And I'm going to give some examples of how studies that kind of come under the umbrella of the BioGeoSCAPES umbrella inform the traits that we use to simulate some really interesting microbial communities. And so I'm just going to use one case study to talk to this because there's not too much time. So I just want to go take a deep dive into this one. And there's been this, to address this question that has been around for a little while and been debated in the literature, which is, and if you don't know this, many of you, it's quite niche, so many of you may not, but why do we find thriving populations of this particular kind of bacteria, aerobic nitrite oxidizing bacteria, in marine anoxic zones? So it's a mouthful, but these nitrite oxidizing bacteria, they're putatively aerobic, meaning that they consume oxygen and they rely on oxygen. But in the last decade or two, we found that they're actually thriving in the most oxygen depleted environments in the ocean. And so the question is why? So why does this happen? Let's remind ourselves of the anoxic zones. So the anoxic zones are these large areas in the ocean where there's little to no oxygen. Everywhere where you see a blue dot here, you're seeing a deep blue dot is essentially an area where the interior of the ocean, part of the interior of the ocean is anoxic. And these areas of the ocean harbor a really unique microbial ecosystem of cross feeders and competitors. It makes for a very interesting ecosystem to study that is still not well understood. So going back to our box analogy of the ocean in terms of a classic biogeochemical model approach, right? So we draw our box and we have our ecosystem component within it. We have some supply of resources. We have oxygen coming in, even though it's depleted. Inside the box, we have organic matter coming in. We've got nitrate coming in. There's aerobic and anaerobic, meaning not oxygen consuming metabolisms within this box. And this is our ecosystem. So we have denitrifiers here in the yellow, we've got nitrifiers in the blue, and we've got anemox in the green. And I think just by looking at this, if you take nothing else away, what you can see is there's a bunch of really interesting interactions that could play out here. We've got cross feeders, so mutualists, in that here we have ammonia oxidizing archaea, which are oxidizing ammonium through to nitrite. And then nitrite is then used by NOB to oxidize through to nitrate. So that's a sort of a synergy. But they're also competitors. So they're competing for oxygen because these arrows are blue and it's an aerobic process. We also have competitors. So there are many different kinds of microbes in this environment that are competing for the same resource, nitrite in this case. So we have the denitrifiers, this nitrite reducing bacteria, which is consuming nitrite. We have the nitrite oxidizing bacteria thriving, apparently, consuming nitrite and competing with them for that nitrite. So this is what they look like, these nitrite oxidizing bacteria that are thriving in this ecosystem. These are the observations that I'm talking about. So these anoxic marine zones contain these thriving populations of aerobic nitrogen oxidizing bacteria, and this comes from a study by, well, it's a integration of many different data sets that was published by this Earth System Science publication by Tang in 2023, where all the data is allocated. But on the y-axis, you've got number of gene copies of this NOB bacteria. And on the y-axis, you've got number of gene copies of AOA bacteria, this ammonium oxidizing archaea, both of which are competing for oxygen in these low oxygen zones. And what I've done here is I've colored the abundances by the oxygen concentration. So from blues to yellows, you're going from very low oxygen to higher oxygen. And what you'll notice is that as oxygen goes down, the NOB concentrations go up in terms of their gene copies, copy numbers. And this is what's telling us that they're thriving in these low oxygen environments. So here is an example of where a BioGeoSCAPES-like data set is being used to inform our understanding of this unique environment. But the interesting thing is that they shouldn't be. So NOB should not be outcompeting AOA in this environment because they're bigger. So I showed you a picture of them before. They're actually much bigger than these ammonia oxidizing archaea. And they shouldn't be outcompeting AOA in this environment because of our theoretical understanding of competition for oxygen. So this is a paper by Emily Zackham, which developed this O2 star. It's like R star from Tillman, if people are aware of it. But O2 star is the subsistence oxygen concentration that's required by an organism to sustain its biomass. And basically the winner has the lowest O2 star. And if you noticed, this is the equation. It has a bunch of terms in it, but one of the most important terms is this R term, R squared, which is the radius. And so if you have an effective radius that's greater than your competitor, this term being in the numerator means you have a higher O2 star, which means you're less competitive. So just based on theory, nitrate oxidizing bacteria should not be outcompeting anything in the low oxygen zones. But what we do know is that they grow very quickly. So we have some estimates from laboratory studies in the red here. On the y-axis is the doubling rate of these organisms per day. So we go from 0 to 5 here, so growing very fast at the top, growing very slow at the bottom. And then we have different organisms on the x-axis. This is our heterotrophic bacteria here. Can you see my pointer, by the way? Thumbs up, yeah, go. And then AOA, which is this ammonia oxidizing archaea here, NOB here from the AMZ, the anoxic marine zone, and NOB from the oxic ocean. So the laboratory estimates that we have suggest that NOB do grow faster than AOA, but they're limited. Not everything can be cultured, right? So we know that many things in the ocean, actually, we struggle to culture things, and that's a big problem. So we take a sample, we can't culture it, we have no idea of actually their traits. And one way that we can get at this is actually using omics. So there's this great correlation between the codon uses bias, so which kind of codons is used by an organism in their genome to select for particular amino acids, and their growth rate. And so we used this particular correlation, took metagenome assembled genomes here that were provided by Shin-Sun, used the model developed by J.L. Weissman to estimate, using this codon usage bias, the growth rates of NOB in the environment and AOA and also Pelagibacter. That's what you're seeing in these black dots. And there's a lot of error around them, but overall you can see the NOB grow faster than the others. And so this is a really good example of where omics can be used to get an understanding of the traits of an organism, a microbe in the environment. And this can be really important, right? So if something's growing faster in an environment, particularly a variable environment, like the OMZs, this is a model representation at high resolution of the low oxygen zone of South America. The light colors are low oxygen. You can see these filaments that kind of work their way into the low oxygen zone and provide whiffs of oxygen. And if we think about our biomass, which we're representing explicitly. If we think about our biomass in terms of going through boom and bust population dynamics, these whiffs of oxygen can provide these this relief to these NOB, which maybe are tracking along, not growing very fast. Their biomass is declining. They get a whiff of oxygen, suddenly they spike. Their biomass spikes, then they sort of die off again, and then we have these boom or bust population dynamics that are possible because we have a high resolution model with lots of filaments, but also we're representing their traits properly because we have this information from omics and what have you, and we're representing their biomass explicitly. And so if we put all this together, here we have oxygen on the left, deep red is low oxygen. In the middle is the nitrite concentration, which is a resource that's required by nitrate oxidizing bacteria. We place this ecosystem, this interesting ecosystem, within a three-dimensional high-resolution biogeochemical model. And these are the rates of nitrite oxidation we find. And as you can see, it just lights up on the edge of the low oxygen zone, where you get this intersection of resource supply, oxygen and nitrite. And because they're growing, well, it's not totally because they're growing quickly, but it certainly helps. It allows them to utilize these resources and temporarily compete for those resources and do quite well. And so this is where we fit all these things into the model. And we can then make, how much time do I have, by the way? How am I going? 1 minute? Okay. Well, anyway, this is where we can then use the models to scale things up to the large scale, right? So we can then ask a question, well, given that we're explaining why nitrate oxidizers exist in these low oxygen zones, what are their effect on oxygen? And what we find using this model is that that represents NOB performing well in these low oxygen zones, like we see in the observations. We see that they actually contribute substantially to oxygen consumption at the edges, up to 50%. So this means that they have a stabilizing effect on the low oxygen zones. If you decrease supply of oxygen, you decrease their oxygen demand. Maybe the OMZs, the low oxygen zones, their geometry, their shape, their volume, maybe they stay constant, even though you're changing the physical supply. And when we actually compare our estimates of NOB's consumption of oxygen to data. So these black dots here on the right are data-based estimates of NOB consumption of oxygen. And when we compare that to what our model predicts in terms of the 25th to 75th percentile of contribution to consumption around this sort of low oxygen zone edge, you find that it agrees quite well with the data. So that's really encouraging and suggests that we're representing the ecosystem well and we're presenting the traits of NOB really well. So all the observations sort of fall within the major quartiles of the data, or maybe within there's some more extreme values here between 5th and 95th percentile, but it's all within the observational bounds. So here's where some rate measurements come in, and just putting it all together and finishing there. We have our biogeochemical models, which we can represent simply to more complex. We have our BioGeoSCAPES like data, and when you put it all together, it can lead to some really interesting insights into how a interesting ecosystem like the low oxygen zones works. Yeah. So thank you all. And yes.
Well, thank you so much. Well, sorry, you'll be the only one. I'm the only one here who was applauding. This was great. This was a great introduction to BGC models and also a very interesting case study. I am looking forward to the discussion. But for now, I want to immediately announce the second speaker of today, which is Professor Sinikka Lennartz. She is a professor at the University of Oldenburg in northern Germany. Her focus in general is on including microbial processes into biogeochemical ocean models. So she's the perfect fit for BioGeoSCAPES. She has work on interaction between microbial communities, emissions of climate-relevant gases, and today she will speak about modeling of dissolved organic matter in the ocean. Sinikka, thanks for being here with us today. Please,
Charlotte, I'm sorry, I'm gonna interrupt by just reminding
yes.
folk that if they want to contribute to enriching discussion after Sinikka's talk, that you should post some questions using the Q&A, and then we'll use those to drive a discussion at the end. Thank you. Sorry for interrupting.
No, perfect. This was the time I needed to sort my screen. Thank you. And thanks for having me. Thanks for the nice introduction. I'm going to jump right in, and you will see some similarities and popping up. So also I would like to start with like visualizing the challenge again that we face of kind of having these small scale processes that happen on the molecular and cellular level, and then ultimately drive global elemental cycles, such as the carbon cycle, which impacts the climate system, which then feeds back to these small scales. So we have a feedback loop that spans more than 14 orders of magnitude in space and time, and that is a challenge for models, as Pearse pointed out previously. So that's from the drop to the ocean. And I will focus now on modeling the remineralization of dissolved organic matter, which I think, so I will present a case study, but it touches also on some points that I think are generally relevant for BioGeoSCAPES in terms of how do we cope with this complexity and diversity. This is highly collaborative work, so thanks to my co-authors, which I have listed here. I was also asked to give a quick intro to biogeochemical modeling, but I think Pearse already did an amazing job in kind of highlighting the main assumptions and processes. I just want to add maybe 2 points, because I often hear that sentence, "Oh, you're a modeler, great, we can put my data into your model." So I just want to differentiate between kind of the statistical approach that starts with data and then identifies a model that represents data, which is a little bit different in the models that we're using. So when I talk about a model, I mean a set of differential equations that tracks how a system changes over time. So we're actually starting with an assumption or a concept, which we translate into differential equation and then run a simulation and then compare to observations. And usually we find some systematic mismatches, so we need to refine our assumptions here. So we spend a lot of time in that loop, but if we're satisfied with the system, we can then start kicking it and asking questions such as, what if we increase the CO2 concentration in the atmosphere? So we can run scenarios. And the basic structure of kind of biogeochemical models is usually the NPZD structure, so standing for nutrient, phytoplankton, zooplankton, detritus, which become the state variables or differential equations, which are then linked by specific rates, which are the terms in the differential equation. But that's just the backbone. So the biogeochemical models nowadays include much more components, have different nutrient cycles, different phytoplankton groups, and so on. So much more details, but they differ a little bit in their setup. And so the question that we often hear is this: what do modelers need? And I think this is also relevant in the BioGeoSCAPES context. And I also don't think that there's one answer, because it really depends on the question. But I've just come up with, from my perspective, 3 points where I think in general we can kind of meet with the experimentally and observationally working people and modelers. And one is the good old process understanding, like what are the key players in the system and how can we meaningfully classify them, because that will become our state variables. And then we need to fill these equations with life, and that is the rate and dependencies. So what are the metabolic rates and what do they depend on? And what is the functional relationship behind that? Is it linear or exponential? Because that describes the terms in the differential equations. And then we need some field observations, preferably like covering different biogeochemical regimes, different times, spaces, which we need for hypothesis generation and also for model evaluation. And then, of course, we have different modeling tools. And I think one way to look at this is kind of in this trade-off of like computational speed or integration speed and level of detail. And I just picked 2 extremes, and they are highly biased by my background. But we have models, as Pearse already pointed out, that have like a high spatial resolution, can model very small details, but also if we think about the Darwin model, which is developed at MIT and McFollows and Tagliabue, they can even model the biogeography of ecotypes of phytoplankton. But I will talk more about this end of the spectrum. Our system models of intermediate complexity. They have, as you see, a much coarser resolution, but they can run much faster. And I'm interested in the soft organic carbon, which has a lifetime of several thousands of years. So that's why my go-to tool is a model that I can run to these long time scales in reasonable amount of times. But there are different tools and they can all kind of excel at a specific part. But what I think is one common thing in all of these models is that they heavily focus on the carbon fixation. So technically, the phytoplankton component is represented in much greater detail than is the counterpart, the organic matter remineralization. So we have different phytoplankton functional types. They require different nutrients, so we model the nutrient cycles, different traits, trade-offs, and so on. And then usually the organic matter degradation is much more simplified, and we often just include this as kind of a rate. And I think that imbalance might be of concern if we want to know how much carbon is stored in the ocean, because that depends not only on how much is going in, but also how much is going out. So this is what I will be focusing today, talking a little bit about why it makes a difference to actually represent bacteria in a model. With bacteria, I mean heterotrophic bacteria and archaea, and I will shorten this down now to bacteria. I'm very sorry for all microbiologists present. And then immediately posing the question, how? And I don't think I can answer this ultimately, but we have some ideas, and I think this is something where BioGeoSCAPES can make an impact. So how do we deal with the sheer complexity, the diversity, and the interactions? So why the soft organic matter? I think it's important, or it's a central part of the marine carbon cycle for two main reasons. One is it stores carbon, 700 petagrams of carbon at the moment. That is more than all living biomass on Earth combined. And it's accumulating in the presence of mainly carbon-limited bacteria, at least in the deep ocean. And this has puzzled scientists for quite some time. But it also shapes primary production because kind of via nutrient remineralization. So it requires an adequate representation in biogeochemical ocean models. But the way this is traditionally done is more kind of a net removal rate approach, which achieves very good agreement with the patterns that we observe today. So the soft organic carbon is then kind of produced by phytoplankton, is channeled into different prescribed reactivity classes that each decay with a first order degradation rate constant. And as I said, this for the present day, these work very good. They achieve very good agreement with observations, but the actual consumers are not really represented in that equation. That means any environmental condition that impacts the ability of bacteria to process the soft organic carbon will not show up here. And so we've recently contrasted that with what we call the MicDoc approach, microbial DOC interactions, where we do not differentiate between different reactivity classes, but the degradation rate is basically a second order reaction as a function of DOC concentration and abundance of bacteria. And if we just plainly put that into a model, this is what we get. A rather homogeneous background concentration of the soft organic carbon, not really reproducing what we're seeing. Here in these lines, these are observations with higher DOC concentrations in the low latitudes, especially in the subtropical gyres. And in a one-to-one comparison, we're not doing very good, which is maybe not surprising because in that model, all bacteria have homogeneous traits. So we do see an accumulation in the presence of carbon-limited bacteria, but we're not seeing the pattern that we observe. And yeah, I just want to mention work by my PhD student, Ben, who looked at the North Atlantic as kind of a case study, because there we find accumulation of the soft organic carbon. And he tested several hypotheses, for example, bacterial nutrient limitation, which we know from experiments, additional production by exudation by phytoplankton, or top-down controls. And he shows that in the model, at least, you really need the factors influencing the consumption side, so the bacteria, and you cannot explain that DOC accumulation purely by a change in DOC production. And so we tried a lot of things. I just want to present one thing which I think fits very well to BioGeoSCAPES because it links experimental approaches with omics approaches with model development. And so here in this study, I compiled, I think, 25 different studies that did nutrient amendment experiments for the heterotrophic community. So taking the heterotrophic community in the surface ocean, adding nitrogen, phosphorus, and organics. And it turns out that in most of these regions, the heterotrophic community is actually limited by nitrogen or phosphorus or a co-limitation with organics. So this is one of these studies which I think provides a very solid database throughout the whole Atlantic Ocean by Hale et al. 2017, showing that in the northern part of the Atlantic Ocean, we mainly have phosphorus limitation, whereas in the southern part, it's mainly organic nitrogen. And this fits very well with omics data on phosphorus and organic nitrogen stress, which was kindly provided by Adam Martini, which also shows higher stress of phosphorus in the northern part and of organic nitrogen in the southern part. So for these omics data, we have a much larger coverage than these punctual incubation experiments. And so if we extrapolate that and then implement nutrient limitation in the model for the bacteria DOC uptake, we get a pretty good fit in terms of the pattern for the limitation factor, but we also, when we reduce the DOC uptake rate by the bacteria, depending on the nutrient concentration, get a pretty good fit after the soft organic carbon concentration on the surface ocean. This was, again, the simulation before, and this is when we tie it to the nutrient uptake. But I just want to point out a few other studies that also now included bacteria in globular system models. One that is, I think, about to come out. So I took it here from bioRxiv, but have a look out for this study by Emily Zakem as well, who really goes beyond that by having different functional types for microbial heterotrophs and really can model the biogeography here. And then one study by Heather Kim, and the study looks at the projected change of the heterotrophic bacteria under climate change. And so I think we now really have, or we're starting to see this diversity in models also on the bacterial side, which I think is very great. But the point that I want to make here is that when we take this traditional net removal rate approach and this MicDoc approach, so where we have an explicit formulation of bacteria in the model, we achieve an almost identical quality of fit with our observational data in both of these cases. And so you might think, okay, then why should I bother with including bacteria in the model? But this actually, I think, shows up when we model future scenarios of the soft organic carbon, because here we're starting basically at the same point, but then diverging into different trajectories. This is work by my postdoc Takasumi, who run future high emission scenarios in this traditional net removal rate approach and the MicDoc approach. And you see here we're having a rather sluggish DOC pool, whereas here we're seeing an increase because we're projecting nutrient concentrations to decline, which would then hamper DOC degradation and lead to an uptake. So I'm not saying that one of these is wrong or right, and I'm also not saying that this is the final answer, but we're seeing differences here. And I think this requires really further research where I think BioGeoSCAPES can make an impact. So I hope that convinced you that we need to explicitly consider these processes in models. But then, of course, how do we deal with that complexity? And in my group, we are mainly thinking about this in two ways. They are not mutually exclusive, and there are probably more. And that is aggregation. So simplifying by grouping into meaningful groups. Of course, the question is, what is meaningful here? And then emergent properties. And we take, in both of these aspects, we take a network perspective. So we're seeing microbial communities and their exchange with the substrates as kind of a network. So the first outlook I want to give is this work by my postdoc Felix Mielke, who took a large data set, 2 transects from the Atlantic Ocean and the Pacific Ocean, and grouped the microorganisms by their co-occurrence and identified these cohorts. So kind of lowering these hundreds of taxa into 10 systematically co-occurring groups. And what I find interesting is that this group composition is stable even in regions that are not geographically connected. So of course, there could be other ways to group microorganisms, for example, by functions. But this is one way of kind of aggregating and grouping. And the other point I want to point out is this question of emergence. So emergence means that the system has properties that the individual parts do not have. And one way that we recently, or in the last years, worked on is that the soft organic carbon persistence can be seen as such an emergent property. So we're now not in a global scale model, but really in a network model where we have different DOC compounds and different bacteria. And the model basically works like this. We have carbon flows from outside the network to bacteria, they diversify it, taken up by other bacteria, they diversify it again. So we have an uptake and release network. And what we end up in this model system, if we translate that into a differential equation, even though all of these compounds are labile, they are available to the microbial community, we end up with a persistent dissolved organic carbon pool with the concentration and radiocarbon age as we observe it in the ocean. So kind of looking at this emergent property. But of course, we cannot, or maybe we don't want to model hundreds of compounds in an Earth system model, so we need to have an aggregation step again. So we use the mean field approach to simplify that network to retain the behavior, but basically have one DOC compound and one box for bacterial biomass, which is basically the model that you saw in the first part of the talk. And we've extended this approach to also include phototrophs because biogeochemical models at the moment tend to focus on purely, or the main interaction between microbes that you see in these models is competition for resources, which I think for phytoplankton, you might get pretty far with that, but probably for the heterotrophic community where we have a lot of cross-feeding and metabolite exchange, we may need a different concept. And this is what we tried with this AMCOM model. This is what my postdoc Leo develops currently and has released here publicly. And the idea of this model is that you can easily, it has a very simple backbone, but you can easily scale it up. So you can use hundreds of different phytoplankton or hundreds of different DOC compounds. And just to show you the kind of questions, we're now at the community level, I think, where this model can make advances. And we can see that this topology matters for the succession and the elemental turnover. So just to show you two examples, very striking, a hypothetic community with very directed interactions looking like this results in a succession that looks very different from a community where everyone interacts with everyone. And then we can apply this to all kinds of questions, and that's what it's made for. We can apply this to incubation experiments. We can switch on and off interactions and look at their role for the competitive fitness. And we can ask stability and resilience questions on this microbial community level. And with this, I'm coming to the end of my talk. I hope I could show you that it really makes a difference if we explicitly include these microbial processes, and that the DOC pool may be much more variable than previously thought if we really go to these microbial timescales. That aggregation and assessment of emergent properties are two potential ways to deal with this complexity, and that we can really link data and model development on different levels. But I think my main takeaway is that we maybe need to shift the mindset from passing the data to modelers towards this kind of joint side-by-side endeavor. And with this, I'd like to thank you for your attention, thank my funders and my group, which I think makes me very optimistic that this interdisciplinary collaboration can work. Thank you.
Great, thank you, Sinikka. Hi, everybody. My name's Alessandro, Tagliabue, University of Liverpool. I'm going to try and manage a discussion in 9 minutes, so we may surpass our end time, but I do want to try and give some time for discussion. So first, thanks Sinikka and Pearse for those really fantastic talks that were really inspiring. I'm going to ask each of the speakers a question based on the questions we had in the chat. I'm going to ask, if possible, to try and keep the answers as concise as possible. I know I'm never adhering to that, so I don't expect you guys to either necessarily, but let's try and do that so we can have more questions. So Jacob Cram asked the first question in the chat, so it would be really rude not to read his one out for you, Pearse. And I don't know if you've managed to see it, Pearse, but he wanted to ask you about how generalizable the results you found in the ETSP were to other low oxygen systems, either in the North Pacific or in the Indian Ocean or coastal margin.
I think they're generalizable because we use a general NOB. So all the traits that we use for the different microbes are quite general. They're not specific to a particular low oxygen zone. And we also tested a lot of different combinations of traits within the sort of realm of possibility that we see in the observations, if that makes sense. And we do find similar results. So yeah.
Brilliant. Thank you, Pearse. Sinikka, you had a question from Kyungmin No, and also quite a similar one from one of our esteemed co-hosts, Charlotte here, which was asking a little bit about the kind of way you parameterize differences between the two different approaches. So Kyungmin was interested in understanding about the timescales and the sort of kind of memories, I guess, of the ocean associated with DOM when you apply these different approaches to the bacteria. And then Charlotte was asking, I guess she was assuming that there was a temperature dependency in one version of the model for the bacterial growth rates, but maybe not for the decay rate. And how does that explain the differences? And so maybe you could kind of combine those questions, if that's okay, to kind of tell us a little bit more about the parameterizations.
Yes. So I think the main reason why we see or I expect larger differences in models where we actually explicitly resolve bacteria is because they operate on much shorter timescales. And the reason in these net removal rate models, we're really talking about net removal. So that means the degradation rate constant must be low enough so that after the ocean kind of circulation of 1,000 times, 1,000 years, there is still 35 micromoles of DOC left. And that means to balance that, there's also a small input only. So I think only 0 point something % of the NPP actually goes into there. And that, of course, means that it's a rather sluggish pool. So if we think in this net removal rate concept, the changes we'll see will be slower than when we have microbes represented there. And I think the question that Charlotte asked about the temperature is a key one. Because what we're seeing at the moment is that we see the highest DOC accumulation in warmest places. So to me, that suggests that at the present, there's something else than temperature that overrides that pattern. And so in my model, there is no temperature dependence. We tried several things. We see, or what literature says is that with like increasing temperatures, we would see an increase in the DOC uptake rate. But the growth efficiency also has a temperature dependence, which kind of offsets that. And so I think this is really a key one where I would be very interested to see also more studies coming on, because I think that temperature question for me is not yet resolved in which direction we're going.
Brilliant. Thank you, Sinikka. I'm going to ask you one more question, Sinikka, from Damien, which asked you, it sounds like a simple question, but there may not be a simple answer, which is like, how many compounds are necessary from your network aggregation to encapsulate an accurate prediction? They're Damien's words, not mine.
Yes, thanks for that question. And I think that is the tricky one, right? How do we actually merge that with data? So the MCOM model as it is now, I use at the moment rather as kind of a conceptual tool. So what constraints on a microbial community topology do I need to reproduce certain observations? We can also kind of really scale it down. And I think I've applied it to an incubation experiment and just had kind of one bacteria strain, one phytoplankton, and then just one DOC pool. But it really depends on the questions, and I think this is tricky to really resolve all of them. So maybe we need to go to an abstraction level of thinking about this as distributions of node degrees or distributions of traits in that system, and then use the model as kind of a more conceptual way to think about this. But I'm very happy to explore different ideas with that tool.
Yeah, brilliant. Thank you, Sinikka. So I'm going to come back to Pearse. I'm going to ask you a question, Pearse, about your really interesting aggregation of the complexity of some of the nitrogen cycle processes there. And it seemed to me that you also needed actually quite a complex physical model, as well as this complex kind of microbial model to get the results that you needed. And I wonder if you had some thoughts about how we might go about integrating those kind of results into the larger scale models that we want to use ultimately for forecasting, right? So it made me think of the fact that, how is this process contributing to the expansion or not of oxygen minimum zones, et cetera, et cetera. But to run those kind of simulations, you can't do that in a ROMS configuration. And so have you had some thoughts about how we can go about bridging that gap, or are we still left at this kind of impasse where we're using very complicated models down at local scales, but we're still left with relatively simple models at larger scales?
Yeah, it's a really hard question. It's a good question, but I think my knee-jerk reaction is we are at an impasse because you really do need the physical complexity. You need the high variability in the environment combined with a complex microbial ecosystem to allow for those winners and losers to develop so to allow for those boom and bust cycles. And it's only the boom and bust cycles that allow for some of these microbial functional types to actually exist and thrive. And so I think you just get a completely different answer if you have a coarse resolution model with no filaments of oxygen coming in. And you'd have a completely different answer if you didn't represent your ecosystem with an appropriate level of complexity in terms of the metabolisms. Yeah, I think we are at an impasse. Maybe we just need to wait until we have some perfect mix of like GPUs, CPUs, and quantum computing comes in, and then we do it all. I don't know. So yeah.
That's cool. That's cool. I think that's a really interesting answer. Sinikka, do you have anything you want to add? It'd be good to finish with a kind of broader discussion, I think, about how do we see ourselves making progress in the sort of ocean biogeochemical modeling community? Are we stuck? Because we still want to make forecasts, right? We want to put a team on the pitch to play those games, to communicate how biogeochemical cycles are going to change over the next decade, century. And what are your thoughts, Sinikka, on how we go about bridging this complexity? I know there's some in your presentation, but I think it'd be good to contrast with Pearse's perspective on that.
Yeah, it is a tough question. I think one of the first things is probably communication between the different disciplines, which I think it's like generating this mutual understanding of how these work processes work. And I think this is something where BioGeoSCAPES definitely can make an impact. And what I really encourage is also seeing these differences in or model diversity, because I think this is something where we can learn and test, for example, which grouping is meaningful for the question that we have, which approach is useful. And so I'm actually quite glad that we're now seeing this diversity also on the heterotrophic side. For sure.
Thank you. Naomi's got a hand up, so I feel like I should let her say something.
No, I just think I really love how you articulated the challenge first. And I think that maybe this is one of the things that we're hoping to kind of tackle head on in these discussions and the in-person workshop. I don't think we're going to come to the answer, but I'm optimistic that maybe statistical approaches or kind of other ways to figure out how to do the upscaling of the high-res physical biogeochemical interactions to the global scale. I mean, the atmospheric dynamicists do these kind of nested models. I don't think we can use exactly the same approaches that they do, but I think the idea is that we have a huge challenge as a modeling community, as you really nicely articulated, and I think there's a lot of potential for us to kind of bring our brains together and tackle it.
Yeah, I did think about something. It's a really good comment because I did think about after I made that comment just before the answer to Al's question, I thought about downscaling. There's some progress. There's a lot of progress in machine learning downscaling of coarse resolution to high resolution that can be useful for all sorts of things, particularly extremes. And those sort of whiffs of oxygen you get in the low oxygen zones, they're kind of like an extreme, right? So
Yeah.
I think machine learning could be really useful.
And that's a note for the attendees because the next one is on statistical approaches and networking, which is next week at the same time. And then the last one is on cellular scale models, which again also think will help us maybe tackle some of these challenges. If you're registered for this one, you are automatically registered for the next two. But spread the word and others can join. You can still sign up.
So I think we'll close there. I think it's 5:03 in the UK. The sun is shining. I don't know what time it is where you are, but I'm sure
Past 4 a.m., hasn't it?
yeah, exactly. Thank you, Pearse, for joining us so late in the day, and all our participants actually on the Pacific Rim, where it's not the most hospitable time zone. We're very aware of that and send our thanks and apologies for the timing. So yeah, I guess to finish up, I'll thank Sinikka and Pearse for two really fantastic presentations. I thank all the attendees for joining. I think it was fantastic to see you all. And to remind you, I guess, that this is a start of a conversation to try and grapple with those challenges that we just articulated, to understand within the modeling community what our options are, what our types of solutions, who's pushing the boundaries, and where might we collaborate with each other, and also with the wider experimental observational community to make progress on these really important questions. So please do try to join next week and the week after, and then to keep a lookout for an opportunity to sign up for an in-person gathering where we can all at least be in the same time zone with differing degrees of jet lag in lovely Woods Hole in September. So I think I'll thank everybody and see you next week. Bye-bye.